Large animals like elephants and blue whales also have a lot more cells than humans do. If every single cell has an equal chance of developing cancer, then why do all these blue whales and elephants not have cancer yet? Hi everyone, I'm Reza and you're watching Scientistica. If you're new to this channel, I'm a scientist by profession, and in this channel I talk about intriguing and paradoxical scientific topics. If you like my videos, please make sure to leave a thumbs up and subscribe to my channel and click on the bell icon to stay notified as soon as I post a video. Without further ado, let's jump into today's topic. When some living cells in our body start growing aggressively, they form a mass of cells called tumors. Tumors can stay benign and harmless, or they can become malignant and start invading other tissues and organs of the body and become cancerous. Cancer usually forms due to some kind of mutations in the gene. These mutations can happen just randomly when the cells are dividing, or they can happen from exposure to environmental conditions like radiation or harmful chemical substances called carcinogens. In 1977, British epidemiologist Richard Peto was studying tumor formation in mice in response to their exposure to carcinogens. He noted that mice that had extended exposure to carcinogenic substances had aggressive cancer formation. This makes sense. As we grow older, our cells divide more number of times and as a result have a higher chance of picking up a mutation and becoming cancerous. If every single cell has an equal chance of picking up a mutation and becoming cancerous, then large, long-living animals that have a lot more cells than humans should already have cancer by now. But this is not true, and this seemingly puzzling scenario was termed as Peto's paradox. There's one theory that suggests that in the case of large animals like blue whales, Peto's paradox might not be a paradox at all. These blue whales do get cancer, but they also get what are called hypertumors. When cancer cells start growing aggressively, it sends out signal in the form of released proteins called TAF, which is its way of telling the body to produce more blood vessels. Sometimes some aggressive cancer cells start growing so fast the body cannot keep up with them and make as much blood vessels as they need. These aggressive hypertumors then start feeding on the normal tumor cells. These normal tumor cells are now malnourished and therefore they cannot grow to a larger size. Scientists use differential equation-based mathematical models to predict hypertumors. I'll leave a link in the description below if you're interested in learning more about hypertumors. Let's look at other animals that have managed to stave off cancer and live longer. Take this elephant for example. In 2015, a team of scientists started scouring the elephant genome to search for an answer to the Peto's paradox. They found that elephants contain extra copies of a particular gene called tumor suppressor gene TP53. They're called so because they protect against tumors by sensing DNA damage in a cell and triggering cell death, thereby killing the potentially precancerous cells. Other animals like the naked mole rat of East Africa manage to stave off cancer by not allowing the cells to come close together at all. Hyaluronan triggers the cell death response whenever cells come close to each other. This process is called contact inhibition. A lot of these animals that rarely get cancer are not very well studied, but there are good researches that are currently undergoing. Joshua Schiffman and his team are currently exploring the possibility of attacking tumor cells by delivering TP53 genes via nanoparticles. Projects like this may open up newer ways to approach and protect ourselves against cancer. Until then, we research more and we learn. If you haven't been living under a rock for the past year, Chances are you have heard of the COVID-19 outbreak of 2019 that possibly started from a live animal and seafood market in Wuhan, China. Animal viruses that can cross over and infect humans are called zoonotic viruses. There have been multiple outbreaks of zoonotic viral infections in the past: SARS, MERS, Ebola, Nipah, and COVID-19, to name a few. Interestingly, bats act as a reservoir host for all these viruses. A reservoir host can maintain a pathogen population like bacteria and viruses while experiencing little to no sickness. So how do bats survive despite carrying so many viruses that cause deadly diseases in humans and other animals? Keep watching to find out. Hi everyone, I'm Reza and you're watching Scientastica. If this is your first time watching, in this channel I illustrate and talk about intriguing scientific topics. If you like my content, please consider giving it a thumbs up. Subscribe and click on the bell icon to get notified as soon as I post a new video. Without further ado, let's jump into today's topic. When pathogens like viruses and bacteria enter our bodies, the immune system can use pathogen-specific antibodies, small protein molecules called complements, and specialized cells like neutrophils, dendritic cells, and macrophages to sense pathogen DNA and protein patterns and destroy pathogen-infected cells. This immune response is often accompanied by fever and inflammation, making us sick. A combination of several existing theories and research results may explain how bats can carry so many viruses without getting sick. First, bats might have an elevated antiviral defense. Bats are the only mammals that can fly. And during this flight, bats' metabolic rates can increase up to 15 to 16 times the normal rate, resulting in an increased body temperature up to 42 degrees Celsius or 107.6 degrees Fahrenheit, similar to a very high fever. The increased body temperature during flight is often beyond the temperature limit which the pathogens can survive. But increased body temperature can also damage bats' own DNA. So bats may have evolved to increase production of DNA damage repair machinery during flight. This also repairs any DNA damage caused by viruses. Research also shows that bats have higher expression levels of interferons, a protein family responsible for antiviral defense. A second theory suggests that bats may have increased immune tolerance to viruses. A research performed by scientists in Wuhan Institute of Technology in China shows that bat immune system has a lower sensitivity to viral DNA or RNA indicated by loss of functionality of a protein called STING, which is involved in inflammatory response to viruses. The dampened expression of STING allows bats to show no inflammatory immune response while carrying high viral load. Unfortunately, recent outbreaks of bat-borne viral diseases have resulted in worldwide culling and destruction of bat roosts. Bats are very important for our ecosystem. They eat harmful insects that damage crops. They are crucial pollinators, and they play important roles in our agriculture. Humans have constantly invaded wildlife territories since the beginning of civilization, and it is natural that viruses spill over from animals to humans. In Bangladesh, several families contracted the deadly Nipah virus in 2011 by drinking alcohol made from fermented date palm sap from trees where bats are known to roost. Similarly, Hendra virus infection in Australia started in a horse farm built near flying fox habitat. COVID-19 started from a live animal and seafood market in Wuhan, China, where pangolins, snakes, and turtles are illegally traded alongside other intermediate virus-carrying exotic animals. To protect ourselves from these deadly zoonotic viruses, Aside from practicing good personal hygiene, it is important to know our places in the ecosystem and have respect for all the diverse species that cohabit this beautiful planet we call Earth. Coronavirus disease 2019, or COVID-19, has resulted in devastating human casualties and worldwide economic recession. During these trying times, concerns among people about protective measures against this deadly disease is at an all-time high. Scientific institutions and companies across the world are already in the process of developing vaccines for prevention of COVID-19. So how are vaccines developed, and how do these vaccines protect us against pathogens? Why do vaccines take so long to manufacture? Keep watching to find out. If you have watched my previous video on bats and coronaviruses, you already know that anytime a pathogen enters our body, our immune system reacts by recruiting an array of immune cells, antibodies, and complements to destroy pathogen-infected cells. Our immune system is composed of two main elements, the innate and the adaptive immune system. The innate immune system acts rapidly after pathogen or vaccines enter the body. The adaptive immune system is activated later and plays a role in long-term protection against pathogens by producing pathogen-specific antibodies and memory cells. Vaccines activate specific receptors called pattern recognition receptors found on the surface of innate immune system cells like dendritic cells and neutrophils. These pattern recognition receptors recognize specific patterns in a pathogen, which in turn results in production of a large quantity of chemicals called cytokines. Cytokines help activate components of adaptive immune system, including plasma B lymphocytes, which produce pathogen-specific antibodies, and memory cells, which establish long-term immunological memory. Which means anytime the body encounters the same pathogen the vaccine carries, the immune system will be ready with a rapid response. Vaccines can be produced using different processes. Live attenuated vaccines contain viruses that have been weakened by growing them outside human cells, for example in chick embryo cells and eggs, or by growing them in low temperatures. These weakened viruses do not cause strong infection, but can induce strong immune response and help establish long-term immunity against these viruses. However, live attenuated vaccines can be dangerous to immunocompromised individuals and to pregnant women due to risk of reactivation and fetal infection. Non-live vaccines can contain whole pathogens that have been inactivated by application of heat, chemicals like formaldehyde, or radiation. Or they may contain certain subunits of the pathogen, like polysaccharides, nucleic acids like DNA and RNA, or toxins released by certain pathogens. Vaccines that contain non-live pathogens are not infectious. Therefore, these are safe for use in immunocompromised individuals. However, inactivated vaccines also do not induce strong immunogenicity, resulting in short-term protection. Several pharmaceutical companies and scientific institutions are working on developing live vaccines against SARS-CoV-2, the coronavirus that causes COVID-19. Johnson & Johnson, Codagenix, and University of Hong Kong are all using live vaccine strategies. Novavax, Clover Biopharmaceuticals, and the University of Queensland are developing subunit vaccines, targeting the immunogenicity of the S spike proteins on the surface of SARS-CoV-2 virus. The spike S protein of SARS-CoV-2 helps the virus enter human body by attaching itself to a human protein called ACE2. Several biotech companies like Moderna and Inovio are exploring DNA and RNA-based vaccines. Vaccine manufacture is a long process, and each vaccine candidate needs to go through many manufacturing steps, toxicology testings, and a series of 3 clinical trial phases before the vaccine is approved. During phase 1 clinical trial, the vaccine is tested on a small group of people to determine its safety, efficacy, reactogenicity, and immune response. In phase 2 clinical trial, optimal vaccine preparation and dosage is determined by testing the vaccine on a much larger group of people of diverse age, gender, and ethnicity. Phase 3 clinical trials are conducted on thousands of individuals to determine the efficacy and safety of the vaccine. These clinical trials can take anywhere from a few months to several years to complete. It is crucial to maintain social distancing in order to flatten the infection curve until a vaccine becomes available. Food is something we cannot live without. Food is a part of our culture, our companion in all occasions. In fact, our ancestors' diet may have influenced evolutionary changes to help us become the humans we are today. Continue watching till the end as we unravel how the ever-changing human diet has influenced the human evolution. Hello everyone, welcome to Scientastica. My name is Reza and I work as a scientist at a pharmaceutical company here in the United States. In my free time, I like to make videos about intriguing scientific topics. So without further ado, let's jump into today's topic. To understand food and evolution, let's look at the first food we consume after we're born, milk. If we look deep inside at the molecular levels of milk, we find a sugar named lactose. To help digest milk, our body needs an enzyme called lactase that breaks down lactose into simpler nutrients that we can easily absorb. Lactase enzyme is produced in large amounts in babies during breastfeeding. As babies grow up and start eating other solid foods, the lactase enzyme production declines, reducing their ability to digest milk. Now, I know what you're thinking. You're not a baby, and you're able to digest milk just fine. That is because a mutation in your gene allows you to produce large amounts of lactase, unlike 65% of the human population. Around 10,000 years ago, in the Middle East, our ancestors began to domesticate livestock. Cattle milk then became a readily available source of nutrition in the otherwise dry climate. In northwestern Europe, due to low sunlight, humans could not produce enough vitamin D naturally, so drinking cattle milk, which is full of calcium and vitamin D, became a great way of improving calcium absorption and preventing calcium deficiency disorders like rickets. These benefits may have led to permanent mutations in the genomes of these groups of humans, allowing them to be able to digest milk throughout their lives, while the rest of the world population remains largely lactose intolerant. In Central Africa, where malaria is widespread, people traditionally consume bitter-tasting plants, and they are less sensitive to bitter taste compared to the rest of us. Chemical compounds that are responsible for the bitter taste belong to a group called cyanogenic glycosides. These bitter compounds are toxic if consumed in large amounts, but can be protective against malaria if taken in moderation. Modern-day Central Africans may have evolved to tolerate bitter taste better because of the anti-malarial effect of some of these bitter plants. Different types of spices are often consumed with food all over the world. These spices often have antioxidant properties, and they help neutralize the toxic free radicals produced in our bodies. People living in hotter climates tend to use more spices because bacteria grow best in warm temperatures, and adding spices can kill bacteria that is growing on perishable foods like meat. Cooking our foods may also have influenced human evolution. Cooking is a process that increases caloric content of foods, eliminates the need to chew food all day, and thus reduces the energy cost of digestion. This means by simply cooking their food, our ancestors saved a lot of time and energy, which could be used for hunting and gathering, or even developing a larger brain. Although our ancestors' eating habits shaped their genomes for the better, in modern humans, the same genes can promote diseases like obesity and diabetes. During certain times of the year, our ancestors would have access to plenty of food, whereas for the rest of the year, the food supply would be scarce. Our ancestors may have adapted to this by carrying thrifty genes that allowed them to store fat and rapidly gain weight during times of food abundance, which later helped them survive when food became unavailable. But in modern times, food is always available and we do not need to exercise to gather food, so these once beneficial thrifty genes can now lead to obesity and diabetes. So how do we avoid becoming obese and diabetic if it is embedded in our DNA? Well, controlled eating and plenty of exercise helps. Recent research also suggests that intermittent fasting may be a healthier lifestyle choice, which is similar to our ancestors' plentiful eating followed by periods of starvation. But that is a topic for another video. By the way, all the information used in my video are taken from peer-reviewed scientific journals. If you're interested, you can find the links in the description box below. If you liked the video, you know what to do. Until next time, I'm Reza, and this was Scientastica.
Deep down by the sea, anywhere we wish to be. When we're loved, we're happy and free. Come together, we're all family.